Good morning, everyone. My name is Chinisa M. Abidin, your presenter today. This research article under cell evolution is entitled Profiling Cellular Diversity in Sponges Informs Animal Cell Type and Nervous System Evolution. This was a work of Mr. Jacob M. Musser together with his team. This was published on September 23, 2021. Before we proceed to the presentation proper of this research, let's first define the words that are important and mostly used in this research. We have metazoans, which is a group of organisms comprising of multicellular animals, endemocytes, which form the outer shell of the sponge's body, pinacocytes, which make up the pinacoderm of sponges, neuroid cells, which had long tendrils resembling those of neurons, choanocytes, which are the main pumping stations of sponge survival, peptidocytes, which express receptive postsynaptic scaffolding genes, and we have pre- and post-synapse, which refer to processes before and after chemical and network communication between neurons, which form the wiring of our nervous system and that of the other animals. The abstract of the study. The evolutionary origin of metazoan cell types such as neurons and muscles is not known. Using whole-body single-cell RNA sequencing in a sponge, an animal without nervous system and musculature, the researchers identified 18 distinct cell types. The team discovered the molecular features of a family of cells that express digestive enzymes inside the water canals, helping the sponge digest any food particles carried by the water. It turns out that these digestive cells express genes typical of the postsynaptic neuron. The data show a communication system that is organized around sponge digestive chambers using conserved modules that became incorporated into the pre- and post-synapse in the nervous systems of other animals. The background of the study. So, sponges represent a basal animal clade that lack neurons, muscles, and gut. They display canals for filter feeding and waste removal and are composed of three tissues: pinacocytes, chambers of choanocytes, and an inner mesohyl. Sponges have the simplest of bodies, harboring a delicate system of canals through which water flows. They are one of the oldest multicellular animals, so they represent the earliest evolutionary metazoan phylum still extant. So it was a surprise when researchers recently discovered that sponges have as many as 18 cell types, including one with hybrid immune and neuronal properties, even though sponges don't have any brain to speak of. Despite their simple organizations, sponges have genes that are usually expressed in neurons or muscles, including components of the pre- and post-synapse, and perform whole-body contractions that flush the canal system and expel debris. However, cells with integrative signaling functions are not yet known. The biggest question is, why look for neurons and muscle cells in animals that don't have them? Well, the researchers knew that sponge cells express their own cellular signals to the cells of the digestive, nervous, and muscular systems of vertebrate animals. As a living fossils, sponges provide the best evidence for the early evolution of the metazoan cells. Spongilla is composed of 18 distinct cell types. Using whole-body single-cell RNA sequencing, the researchers conducted a comprehensive survey of genetically distinguishable cell types in 8-day-old freshwater desmosponge Spongilla lacustris. To their surprise, 4 capture experiments provided 10,106 S. lacustris cells expressing 26,157 genes. Interrelationships of cell types and sponges. Animal cell types are organized into families, which share expression of regulatory and effector genes and are identified by phylogenetic analysis. This revealed 28 distinct gene sets delineating individual cell types and cell type groups, which hinted at the presence of hierarchical organization. Endemocytes are contractile. The family of endemocytes covers and shapes the sponge body. They include incurrent pinacocytes, apindopinacocytes, and basopinacocytes. Endemocytes are found to express contractile cell master regulator serum response factor or SRF and CSRP123, also known as muscle limb protein, which suggests a conserved regulatory module for actomyosin contractility. Endemocytes respond to nitric oxide signaling. In the marine Dismo sponge, Tethya wilhelma, NO induces and modulates whole body contractions, but only affected the osculum in the freshwater sponge Iphidacium weyleri. During the S. lacustris contraction cycle, the collapse of osculum and incurrent systems first causes excurrent canals to swell, which subsequently shrink to expel water through the osculum before returning to the resting state. The researchers tested the response to NO by treating juvenile sponges with oxadiazolokinoxaline, an inhibitor of NO receptor. NO release immediately shrank the incurrent system, including current canals and tent, and expanded the excurrent canals as during the initial stages of the contraction cycle. Subsequently, the incurrent system exhibited short, pulse movements and returned to resting state only after NO removal. This established the key role of NO in initiating and regulating the contraction cycle in S. lacustris. Digestive peptidocytes express postsynaptic scaffolding genes. The family of peptidocytes share a role in digestion, which is reflected by phagocytic vesicle and lytic vacuole gene expression. The guanocyte subclad is enriched for genes of the receptive postsynapse complex in animals, with neurons including postsynaptic scaffold and dystrophin-syntrophin signaling complex. Peptidocytes specifically express KLF5, which controls cytodifferentiation of intestinal epithelia, and guanocytes express NKX6, RBPJ, and Notch that specify pancreatic cell types and absorptive enterocytes. Communication between neuroid cells and guanocytes. Neuroid cells exhibited a secretory profile via upregulation of the signal peptidase complex and enrichment for presynaptic gene sets, also observed in endemocytes, suggesting communication via vesicle secretion. Notably, nearly all neuroid cells were associated with guanocyte chambers, with most chambers containing at least one neuroid cell in the middle of the chamber or in its lining. The observations were: first, one neuroid cell positioned centrally and another near the apopillar pore, each with multiple protrusions directed toward different guanocyte colors and nearly always contacting and enwrapping one or more microvilli. Second, numerous secretory vesicles 50 to 500 nanometer in size throughout both neuroid cells, and two Golgi stacks per cell, each oriented toward the cellular protrusions. Then, third, both neuroid cells formed long invaginating pockets that encased cilia tips with shaft-like constrictions. These observations suggest a dual role for neuroid cells in intercellular communication and in the clearing of bacteria or cellular debris within guanocyte chambers. For the materials and methods, we have Louvain graph clustering, used for clustering the variety of cell types with distinct expressions, signatures in 2D space, as you can see with the picture provided. Then partition-based graph abstraction, used for exploring developmental relationships, which assesses the degree of connectivity among clusters, given in the picture above. Then we have single molecule fluorescent in situ hybridization, used for imaging cell clusters of selected marker genes, also provided with the picture below. Then we have focused ion beam scanning electron microscopy, or FEBSIM, which is a two-step imaging strategy, a combination of confocal light microscopy and synchrotron-based X-ray imaging, used to further characterize the neuroid cell interactions. Then we have correlative X-ray electron microscopy, which is a combination of laboratory-based X-ray microcity with the FEBSEM data, which allowed the visibility of up to 50 nanometer in size neuroid cells, including in the cell protrusions. Then we have the single-cell RNA sequencing or single-cell transcriptomics, which examines the gene expression level of individual cells in a given population by simultaneously measuring the mRNA concentration of hundreds of thousands of genes. A notable feature of gene and species evolution is a tree-like history of common descent. The researchers observed significant hierarchical organization among differentiated cell type expression programs in Spongilla. They propose that this reflects evolutionary relatedness rather than homoplasy, and that major sponge cell type families such as endemocytes and peptidocytes originated via cell type diversification. Despite the simple organization of sponges, they have genes that are typically expressed in neurons or muscles, including pre- and post-synapse components, and perform the whole body contractions that clean the channel system and expel waste. Their gene activity pattern suggested they were secreting chemicals similar to those neurons released at synapses to communicate with their neighbors. In Spongilla and the calcisponge Cyconciliatum, pinacocytes specifically express the sole sponge ortholog of MSX, which is known to specify sensory ectodermal regions and myocytes in other animals. MSX activates atonal, which plays a conserved role in mechanosensory neuron specification and demarcates sponge pinacocytes together with its binding partner TCF4E1247. Most notably, apindopinacocytes and incurrent pinacocytes express the single sponge orthologs of the PAX and 6 homeodomain transcription factors, respectively, that also play conserved roles in sensory cell and myocyte specification. In endemocytes, experiments revealed coordination of the contraction cycle via nose signaling, which also triggers endothelial muscle relaxation in vertebrates. Endemocyte transcription factor signature support in evolutionary link between sensory contractile systems and sponges and other animals. In peptidocytes, analysis of cellular transcriptomes suggests digestive functions. Peptidocytes thus may have existed in metazoan ancestors, initially with intracellular digestion as observed in sponges and their unicellular relatives, and then acquiring external digestion, first as part of a digestive mucociliary sole and then incorporated into the gut. In neuroid cells, data suggest communicative functions within choanocyte chambers. Their close physical interaction with choanocyte microvillar collars and cilia, the expression of secretory genes, and the presence of secretory vesicles suggest a coordinative role in feeding, possibly by modulating ciliary arrest. Now, for the conclusion of this research. The investigation has just revealed how the different types of cells of these animals communicate with each other to make digestion, for example, ordered by neural signals. This communication between sponge cells may be similar to that of the primitive invertebrates in which the first nervous system evolved. All three cell types families use conserved neural gene sets. This parallels the wider occurrence of neural modules across epithelial and fiber cells in the symbol placozoans and showcases possible parallel routes toward neuron evolution. In line with this, the contractile network and neurosecretory network hypotheses envisage the origin of neurons in distinct cellular contexts that resemble endemocytes and peptidocytes. Their work thus puts sponges a center stage in elucidating nervous system evolution. I think that would be all for this research presentation. This has been Janisa M. Abidin, and thank you for listening.